This is your pre-lab video for the chemistry of acids and bases. Our objectives for this lab are several-fold. The first of which is to determine experimentally Kw, which is our equilibrium constant for the autoionization of water. We'll also be determining the Ka for the ammonium ion. We'll be observing properties of buffer solutions and demonstrate the pH changes that occur during the course of two different types of titrations, as well as demonstrating the connection between the pH of a solution and the color of two different acid-base indicators. I highly recommend you read the lab thoroughly, especially the first several pages. It gives a great, concise summary of all of the information on acids and bases that you need, including the definition of acids and bases as proton donors and acceptors, how water can act as an acid or a base, and the concept of pH. So you've learned this in lecture, that the pH is the negative log of the hydronium ion concentration, or that the hydronium ion concentration is the log base 10 of the negative pH. It goes over the concept of a strong versus weak acid and base. It goes into depth about equilibrium constants and Kas and Kw in particular, discusses the relative strengths of weak acids using the Ka and the pKa, discusses how to determine the relative strengths of weak acids using their Ka or their pKa, talks about what a buffer is and how to make them, as well as titrations. And we're going to go over buffers and titrations separately and a little bit more in depth. Buffers. Buffers are prepared from a weak acid and its conjugate base, or from combining a weak base with its conjugate acid. And what this does is it sets up a solution that will resist change in pH when a strong acid or a strong base are added to it, or when it's diluted. So for example, if we look at the equilibrium of acetic acid, we can demonstrate this concept. So if you have this equilibrium and you have combined an amount of acetic acid as well as its conjugate base, and you put these together, you have excess conjugate base as well as excess acid in the solution. Now what does this do if we add a small amount of strong acid or strong base? We'll take a look at that. A neutralization reaction occurs. If we have excess acetic acid in the solution and we add a small amount of base, normally, without it being a buffer solution, adding OH ions, hydroxide ions, to a solution would certainly affect its pH. However, if we have excess acetic acid around, then it can neutralize the hydroxide ions that were added and just generate more of this conjugate base, effectively removing the added OH ions, and the pH then doesn't change. Similarly, if we have excess acetate ions in the solution and we add a small amount of strong acid, the acetate ions are in excess enough to neutralize the added hydronium ions. Given that these Ks are both quite large, both of these reactions essentially go to completion and will neutralize the added acid or base. So this pH stays nearly the same. It does change a little bit, but it stays nearly the same. And the effect of the addition of this acid or base has been buffered. The pH of a buffer system depends on the ratio of how much acid and conjugate base are present, as well as the Ka of the acid that's being used in the buffer. We often use a technique of titration to follow quantitatively a reaction of an acid with a base. And in this experiment, we will be titrating a weak acid with a strong base with the following objectives in mind. We want to observe the changes in pH as the acid is consumed by the base. We also want to take a look at the shape of a typical titration curve, which is a plot of the pH versus the volume of the base added. So as this titration progresses, how does the pH change as we add more and more base? We'll also be able to determine the pKa of this weak acid that we're titrating. And we also want to take a look at the color changes of various indicators as they relate to pH. And as the pH changes, how does the indicator change color? Here's an example of a titration curve of a weak acid. Now, if I didn't tell you this was a weak acid and you just had this curve, you could certainly figure out that it was a weak acid. First of all, look at the starting point. Before we add any strong base at all, the pH of the solution is less than 7. That means it started out as an acid. As we add base, it increases in pH. We have a sharp change in pH here, and then it levels out again. So what's important about this curve? One is where it starts. The second is this equivalence point here. And the equivalence point is right in the middle of this sharp pH change. And this is the point in which the acid, all of our acid, has been completely neutralized by our additional base. And this is marked by this sharp change here. This is called the equivalence point. Halfway between where we started and the equivalence point is called a halfway point in the titration. And this is the point at which we have equal amounts or equal moles of our weak acid as well as our conjugate base. And this is a special spot because the pH of that point equals the pKa of the weak acid that we're titrating. And this is because of that equal number of moles. Right around this region is what's considered the buffer region. And the reason that it's a buffer region is because we have present our acid as well as our conjugate base. Another type of titration you'll be doing in this lab is a titration of a polyprotic acid. This means that we have more than one ionizable hydrogen. So in this case, this is just an example of a diprotic acid, sulfurous acid, and it has 2 ionizable protons here. And you can tell this is a polyprotic acid because number one, we start below 7, so it's definitely an acid, and then you have 2 step changes. So this is the equivalence point for the first hydrogen that's being ionized, and this is the equivalence point for the second ionizable hydrogen. You have 2 different halfway points here. So halfway between the starting point and the equivalence point is the halfway point for the first ionizable hydrogen, and that's the pKa for the loss of the first one. And then we also have a halfway point for the second. Throughout this lab, you will be measuring pH using a pH meter, and it is equipped with a glass electrode. This electrode has a very sensitive glass membrane that can exchange protons with the solution. And this transfer of charge is what gives rise to a very small electric potential difference, and that's what's being measured by the instrument. The potential difference is then displayed in pH units that will change depending on how much hydronium ions or the hydronium ion concentration that's in solution. Before you start, and as well as in between every single measurement that you make, you need to rinse the glass electrode thoroughly with purified water. And I recommend using your squirt bottle with the purified water into a waste beaker, and then you'll gently remove the excess water with a clean Chem wipe. It will be necessary to stir the solution with the electrode until the meter reading stabilizes. So don't just read and record the very first pH that you see. Stir the solution with the electrode, give it a little bit of time to equilibrate, and let it stabilize, and then record your pH. You're going to get better results if you measure the pHs of the solutions in the order listed in the table in the manual, so make sure you do that. Don't just set your electrode down on the bench in between readings. You need to keep the electrode upright, and you want to store the electrode in solution, so that glass membrane should be submerged in solution in between readings as well. And last but not least, please be very careful of the electrode. This glass membrane is very, very fragile. So as you go through this procedure, and I'm not going to outline every step or even go through the calculation as they are very detailed within the manual. So you need to measure all your pHs and go through all the steps that are in the lab manual. Complete the calculations that are outlined in the report form. So there is a report form at the back of the lab in the lab manual. Please, when you hand this in the following week, you will not have to hand in a formal written lab report, but this report form needs to be complete, it needs to be neat. Please use complete sentences, speak like scientists, and do a nice job on this because you do not have to type up a formal written lab report. So put some effort into the lab report form.